Okay, so our last speaker for today is Ashery. Sorry for the session. Ashery will be telling us about developing a biological definition for Synucleinopathies based on Alpha-Synuclein. I'm going to let you read it. The font here is tiny. Great.
Working? It's working. Thank you, Itai, for running the center and for giving us the opportunity to present our work, which will be on synucleinopathies, and I'll explain to that in a minute. Let me just begin my talk with mentioning that today is the 600 days that we still have 58 hostages in Gaza, and our government is doing everything they can to continue the war and keep them there, and we should do everything we can in order to stop the war and call to release the hostages. We have Atzeret at 5 o'clock at Antin Square. You're all welcome. We'll start Antin, the Atzeret at the square, and then go all the way to the Kikar HaChatufim V'Shar Begin. This is the minimum we can do, and we can do more than that, and hopefully they will be released soon. So with that, I will go back, like in life, from demonstration to science to our project, and this goes to different types of pandemic that belongs to the neurodegeneration. And I'm going to speak about a series of diseases called synucleinopathies and our direction to try to identify it biologically. So first, what is, that's it, no, other side, yes, you're right, sorry. What is synucleinopathies, and before that? So we know that in many neurodegenerative disease, we have a process in different cells that a protein, which has a physiological function, will start to aggregate and form clusters, aggregation, plaques. It comes in different flavors. So for example, we have in ALS, TDP43, we have in Alzheimer's disease, tau and A-beta. In Parkinson's disease, we have alpha-synuclein. And as you can see in many other diseases, Huntington, we have huntingtin. In all these cases, we have a situation where protein start to aggregate, to form aggregation, which are big. Sometimes they are kept in the cell, or sometimes they are secreted outside of the cells and cause plaques or even propagate through this process. And what we don't know at the moment in many of the diseases is which one is the toxic one. We know that this is the physiological functional monomer. It sometimes works as a dimer or tetramer, but we don't know where the toxic function start. And it's different from different type of diseases. So I'm going to speak about synucleinopathies. And for that, let me just explain what is synucleinopathies, and I'll just mention a few diseases. In all cases of synucleinopathies, we have a protein, which is called alpha-synuclein. And alpha-synuclein is a small protein that is sitting in the synapse and also in the nucleus, which is important. And in the synapse, it's known to activate or to be active in the synaptic vesicle cycle. At some point, the protein starts to aggregate. Usually it can work as a tetramer, but when it starts to aggregate, sometimes because of mutation, sometimes because of unknown reason, and sometimes because of overexpression, you get the oligomer, protofibril, fibrils, and then usually in Parkinson's disease and dementia with Lewy bodies, it's also kept in neuronal, intracellular Lewy bodies, which are huge structures composed of membrane, composed of proteins, many types inside the cells. And this causes the neuron to die. So this is the general aspect of synucleinopathies. And we have two main types of synucleinopathies. One which I mentioned, which is a Parkinson's disease. Sorry. One is the Parkinson's disease, and together with dementia with Lewy bodies, we have aggregation in Lewy bodies in neurons. Another type of synucleinopathies is called multiple system atrophy, and in this case, the same protein is starting to aggregate in oligodendrocytes, which are the myelin forming cells in the brain. It also propagates to the neurons, but the main feature is in the oligodendrocytes. So this causes glia cytoplasmic inclusion, and these are Lewy bodies, the same protein, aggregates, and cause cell death, different types of cell deaths in different part of the brain. And MSA, or multiple system atrophy, is rather rare disease. Only 0.03% of the population are sick, but it's very aggressive, and within 8 to 10 years, usually it causes death. Parkinson's disease is the second most late onset neurodegenerative disease, and you have 3% of the population above 65. And dementia with Lewy bodies is also the second most common neurodegenerative disease after Alzheimer. And what is known is that the clinical manifestation of the diseases are very similar. In many cases, you have Parkinsonian, you have autonomic dysfunction in several cases, and this leads to a problem, since although the synuclein aggregation occur in different brain area, in MSA, PD, and DLB, there is overlapping clinical symptoms in both of them that is one of the biggest obstacles in accurate diagnosis. So when you go to the doctor in early phases of the disease, in many cases, they won't be able to tell you if you have MSA or PD or DLB. And therefore, what we are trying to suggest, not only us, but also the field, is that really a time shift from a clinical to a biological definition of PD, DLB, and MSA. And the problem is that the aggregation of synuclein in the prodromal stages of the disease is invisible. And we need to find a way to see it. And basically, we want to develop a biomarker based on alpha-synuclein aggregation, hopefully in early stages of the disease, and with that be allowed to diagnose the disease early and also track the disease progression and also track the treatment, which are currently starting to be available. So I'll start with a short introduction about Parkinson's disease, and then I will come back to the other diseases. So we all familiar with Parkinson's disease, which is the second common neurodegenerative disease. It increases the prevalence after the age of 60, and it's characterized by motor symptoms like bradykinesia, rigidity, tremor, gait, but there are also a lot of autonomic dysfunction, including swelling, including sweating, including fear, including constipations and other problems. 95% of the diseases of Parkinson's disease cases are non-genetic. Although when you have mutation in one of the genes like LARC, PARC, PINK, GBA, and alpha-synuclein, it increases the chances of having the disease, but many cases, or most of the cases, are called idiopathic PDs. Maybe GWAS in the future will tell us some genes that are not expected and they cause the disease, but at the moment it's not known. Most the characterizing feature in Parkinson's disease that also lead to the motor symptoms is the death of dopaminergic neuron in the substantia nigra. And their death caused the Parkinsonian symptoms because there is imbalance of the motor activity. However, in the stage where you are diagnosed with PD, 50 to 80% of the dopaminergic neurons are already dead. So this is a bit too late. So if I look at this scheme here that show the times in years, and this is the diagnosis stage according to the severity of the clinical symptoms. At this point, about 50 to 80% of the dopaminergic cells, which are shown in green, are dead. It is a bit too late to start to treat the disease. However, it leaves 5 to 10, maybe 15 years before this stage that we can maybe find the disease or diagnose the disease and start the treatment. And if I'll try to look in more detail on this graph, I want to add another feature to it. So when the neuronal cell die, at this stage we have already quite a lot of accumulation of alpha-synuclein, which I mentioned before. And if we look years before, the idea is that alpha-synuclein is start to accumulate in these neurons, specifically in Parkinson's disease, years before the neurons start to die. So this leaves us more years, maybe 20 years, 25 years, that we can track the aggregation of alpha-synuclein, try to prevent it, and this might prevent the cell death and also Parkinson's disease. But we have several challenges. First is to detect the aggregation of alpha-synuclein outside the brain. We don't want to do biopsies to everybody at the age of 30 or 40 to see whether they have aggregation, and the imaging technique at the moment cannot detect alpha-synuclein early stage of aggregation. The second is that we need basically to be able to track the transformation of monomeric tetrameric form of alpha-synuclein to oligomers and fibrils and Lewy bodies. So when we have these two challenges met, we might be able to diagnose early the disease, to track the disease progression, and also examine the efficiency of treatment. So how do we tackle these challenges? So nature said that alpha-synuclein is a synaptic protein, not only in the brain, but every synapse that innervate our muscles, our sweat glands, have synuclein at the end. And it was found in the last years that alpha-synuclein aggregation can be found in many other periphery systems, in the skin, in the CSF, and many additional tissues. And what we are looking at the moment is at the skin and at the CSF, and I'll come back to that in a moment. So with that, we might be able to track alpha-synuclein aggregation at early stages or late stages outside the brain. So how do we detect the changes from single to tetrameric form of alpha-synuclein? For that, we are using a technique which is called super-resolution microscopy. For that, we try to lower a bit more the... That's much better. So when you take a confocal image, due to the diffraction limit of the microscope, you will always see some blurs surrounding your protein of interest. Even if you have 1 or 2 molecules here, it will look like 2 to 300 nanometer big structure. What we achieve using a super-resolution microscopy, and specifically this term, which I will elaborate in a minute, is that you can detect a single molecule, and you can detect that here there are 2 molecules, and here there are maybe 20 or 200, and they are at density of X, Y, or Z. So how do we do it, or how do the system does it? We're using a microscope that exists in the, I think it's now called the Gray Imaging Center in the medical center. And what, because the fluorophore, instead of all of them shining together and giving us a very blurry image, we cause them to blink stochastically. So each time we are doing many, many frames, different groups of molecules will blink. And each one of these blink, we can now fit a PSF, a point spread function, and accurately define the center of this blink as the main center of this molecule. And what we are doing is repeating these images many, many times, thousands of times. Each time we are marking the center of the blink, and we are reconstruction a new image, which will basically compose of the different blinks. And this is why it's called direct stochastic. It is the stochastic blinking. Optical reconstructing microscopy, this is the reconstructing itself. So basically, this is how an image look like at the end. And in a way, I'll show you here another movie that shows you a bit more detail. These are blinking molecule that you can see slowly accumulating here. And here come now a second color that you can do 2 colors. And basically, you can create an image, very nice image. In a way, you can say that this is a modern punctualism, similar to maybe what George Seurat did 100 and something years ago. But for us, at the end, this is our end product. And we need to do some sense out of it. So we are developing Ophir in my lab, is developed a lot of cluster analysis algorithm. At the end, she wanted to be able to say that this aggregate has X molecules and the density is Y and the size is F, and then we can compare between different conditions. So this is the system, so we can practically check aggregation outside the brain. We have a system that can detect a single molecule, and initially we decided to do a preclinical study to see that the system is basically working, and in collaboration with Maria Grassa Spilantini in Cambridge, she developed a mouse line which has Parkinsonian-like feature. They express the truncated form of alpha-synuclein, they have excess aggregation, they have motor symptoms, and I will not go into the details. They have loss of dopaminergic neuron, they have loss of dopaminergic excretion, and also motoric system. So what Dana Bar-On did at that time, she received the brains from these mice at different ages and basically tracked the appearance of the synaptic protein alpha-synuclein, an additional synaptic protein. And as you can see here, just without any quantification, at 2-month-old, 6-month-old, 18-month-old, you can see that the organization of the alpha-synuclein in the synapse is different. What we found is that now if we quantified the changes as the disease progress, you can see that when the disease starts, there are less numbers of aggregate, and when the disease progresses at the age of 6 and 12 months, there are more small clusters, and these clusters are clusters that cannot be detected with confocal microscopy, since all of them are below 300 nanomolar. Roughly 250 would be the limit of detection in confocal microscopy. So with that knowledge that there is a correlation, not more than that, between the aggregation and the disease progression, we also collaborated with Christian Giesinger and Armin Giese, who developed an inhibitor of alpha-synuclein aggregation. And when these mice were fed with the inhibitor is called ANLE 138B, and at the moment it's also in clinical trial by Modag and Teva. We found that the mice have much less Parkinsonian symptoms, the neurons did not die, and when we looked into the striatum of these mice, we could clearly detect the cluster, the aggregate are still there, but they are much less dense. So it seems that the modulator is interfering with the aggregation process, and this is the ANLE is now called embolsolmin, and it's in clinical trial for MSA, which I'll come back to that in a minute. So using this term and advanced analysis, we identified specific aggregate subpopulation that correlate with PD progression in a mouse model for PD and show the efficacy of ANLE 138B to inhibit the PD pathology. So with that, we decided to go to a more human-related study and to see whether we can detect alpha-synuclein in skin biopsies for Parkinson patients and compared to control. And the idea here is that we are receiving, in the skin itself, there are sweat glands that are innervated by sympathetic neurons. There are also hair follicles, which are innervated by neurons. And it was shown that these neurons will have aggregation of alpha-synuclein. It was still not diagnostically approved. So this is the work of many students, starting with Shir and Noah Barak and Daphna Fischel, and now Ophir Sadir is running most of the research with the help of Irit Gottfried, my lab manager. And what we are receiving is skin biopsies from Hospital Meir, Tel HaShomer, and Soratsky. We are staining these with different antibodies. And this is how a sweat gland, that's it. This is how a sweat gland look like. And you can see at the periphery, there are aggregation of alpha-synuclein, and we always check for the existence of a synaptic protein, just to be sure that we are looking at the synapse itself. And this is an example of how it looked like in 3D. So these are two proteins that we are imaging, and you can nicely quantify the number of protein in each one of these clusters. For this study, we had only 7 Parkinson cases and healthy control. We are now collecting more cases, but even from these numbers of low number of patients, we could see that in places where we have more pathological form of alpha-synuclein, I didn't speak about it, but it's a phosphorylated one, we have less neuronal tissue, and this is also shown previously. And what was also interesting is that we found that in Parkinson disease patient, we have more aggregation, and this is an illustration of the different distribution of more aggregation of alpha-synuclein compared to control. What we are doing now, and for that we need more, probably much more patient, we are trying to correlate the molecular aspect that we have with the clinical severity of the patient. So just to show you one of the correlation that we did, again with 6 patients, so it's really nothing, and we compared the MoCA, which is equivalent to the cognitive ability of the patients, and to the number of the localization per cluster, and we found that there are different correlation for Parkinson's disease patient and for healthy control, and we'll try to do it for many more patients, and by that hopefully be able maybe to predict also the severity of the disease. So I'm going to shift now to a biological definition of MSA, PD, and DLB based on this system. We have still some time, or yeah, I think we are okay. Okay. So we are doing it in several directions. One of them I'll just show you briefly, and one of them I'll elaborate a bit more. We are trying to compare alpha-synuclein aggregation in PD and MSA model. And by that, what we are doing is we are looking at neurons expressing alpha-synuclein or oligodendrocyte-like neurons that are expressing alpha-synuclein, and trying to learn about the aggregation itself and also about the effect of different inhibitor. So this is one project that is on progress, and also we are trying to look at iPS cells derived from patients with PD, MSA, DLB, and healthy control, and try to understand the different organization of the protein in these cells that might give us more information about the aggregation themselves. But what we are doing, and it's a bit more advanced at the moment, we are trying to compare alpha-synuclein assemblyon in alpha-synuclein seed amplification assay. So I need to explain a bit more about this last sentence that I checked. So Creutzfeldt-Jakob, prion disease, mad cow disease, we are all aware of it. We know that there is a protein that propagates from cell to cell in the brain and causes aggregation of the nearby protein in nearby neurons. And seed amplification assay, which I will describe in a minute, is already in the clinic in order to diagnose prion disease. The status of synucleinopathies is still way behind, but it's getting to it. So what is seed amplification assay? The idea is that if you take a sample, CSF sample, brain sample, skin sample from a patient with seeds of alpha-synuclein or prion, and you mix it with monomeric alpha-synuclein, one of the things that will happen after several rounds of heating and fragmentation is that the monomeric form that you added to the seed will start to elongate and propagate and create fibril. When they create fibril, you break them, you fragment them, and this you run it several times. It's like a PCR for seeds. And if you add a fluorescent tag, it will bind in between the beta sheet, the theoflavin, which you probably might know, and the fluorescent will increase. So this is a method that is used for Creutzfeldt-Jakob, and it's being now tested for Parkinson's disease and MSA with very nice result, which are shown here. So the idea is that if you have a control person without seeds and you put alpha-synuclein, nothing will happen here, and you will end up with a flat curve. If you have a Parkinson's disease patient or MSA patient, you will have an increasing curve. What several labs found is that you can diagnose between MSA patient and Parkinson's disease cases based on this different profile of seed amplification assay. So this is a profile that is for CSF taken from Parkinson's disease, and this one is for MSA, and this was in control. So it's really nice, but nobody knows what's here at the end point of this aggregation. So what we decided to do is to contact some of the labs that are working on it and ask them to send us the end product of the seed amplification assay of PD, MSA, and DLB. And this is something that we are doing with Claudio Soto and with Kai Drakes, who provide us with different types of antibodies. And what we found is, first, if you look at healthy control, so again, healthy control will have the monomer that were there. Indeed, what we found in the super resolution microscopy is that many dots here, I'm not sure that you can see them, but there are dots here, nothing more than that. But when we looked at the assemblyons coming from the different diseases, we saw completely different image or structure, and this is the structure that we got in DLB, MSA, dementia with Lewy body, multiple system atrophy, and PD. So they look different, and this is by staining with a protein against total alpha-synuclein, and when we stain them with a protein against oligomeric alpha-synuclein, we got slightly different staining. And the phosphorylated protein is not working here since these are in vitro. So with that, what we can do, we have a list of patients that we received a CSF sample from them. Hopefully, we'll also get brain samples. And I'll skip that. And you can also see this is just a gallery of many cases. So MSA patients look very similar to each other. PD look different, and DLB look different from the rest. And what we can do now is apply our algorithm analysis and can get different combination of aggregation at different types of the diseases. When we look at the ratio of the aggregation of the oligomeric and the total, we can already start to see differences between the different diseases. Again, this can be added to what is known in the clinic, and this is something that we can also track later on when the disease progresses or when there is a treatment given to the patient. In addition, when we looked at the monomeric form of alpha-synuclein, we can identify and quantify and characterize how monomers look like in the system, and basically say that the monomer for us has a 20 blinking, the size is XYZ, and then we can go back to these places and maybe suggest that we have here oligomers of 20 or 30 or more points. So there are structural and organizational differences between the different synucleinopathies, and this is something that we are proceeding, and I'll just summarize. So we have a user-friendly quantitative analysis platform that can offer a quantitative aspect for, in this case, alpha-synuclein, but any other protein of interest that you are working on, cellularly or in tissue. One can quantify with this form and get a lot of information about the structure of the organization, the number of molecules, and many other information. We provide with this term molecular differences between healthy control and Parkinson's disease patient, and we have preliminary results suggesting structural and organizational difference between different synucleinopathies. And with that, the platform, basically, we can use it working with individual at-risk population, carrying GBA mutation, for example, which can track disease progression and also efficacy of treatment. And just to thank the people who did the work, and this is a work of many, many people since they are involved in hospitals. It's supported by Michael J. Fox, but Dana did the initial work with the mice, and Ophir did most of the work that I presented on the patient and also on the DLB and the synucleinopathies, and they'll be happy to answer questions. Thank you.